Hi, I'll be walking us through the demo of bees. So, what we have here is a PC standard specifications as a very simple 6 core processor along with 16 gigabytes of RAM, and there is no inbuilt, I mean, there's only the inbuilt GPU. We don't have an external GPU for this particular PC. Closely represents the general systems that are available. Along with that, as you can see, the 8 GB, the image, which is the offline annotation engine, as well as the offline databases, all are integrated in this 8 GB, including the annotation engine. And along with that, we also have the LLM model, which is of 3.3 GB and is also present in the PC. Now, what we will also do is now very quickly, since we have launched it, we'll go to localhost, and we shall very quickly get to the login page. Now, as we can see, the page is maintained in a very standard way where login and passwords are quite standard, right? Because it's HIPAA compliant. So we're making sure that the session that we have and the roles and also the different users, there is like data security ensured. As we can see, this is the general web page that loads up. It has all the details which we currently need uh, in order to look at the overview of the different cases present. As you can see, how many analyses have been done, what kind of cases were there, and a very simple plot. So the way we add new cases are, let's say we try test, let's say 10, for example. Let's say this patient is of like 48 years old. Let's say it is a male. Let's look at, so currently it supports both Ensemble and RefSig databases. So for now, let's say we use the RefSig database. For now, we are only supporting GRCS 38, no, not supporting GRCS 37 yet. But definitely, we intend to add this. Now, the sample that we have is of gastric cancer. And as you can see, the DOID based approach is taken so that one can like add the cancer type very easily. And of course, there is a quick what do you say, disclaimer saying that there is no personal information that is included, like no directly identifiable information. And let's say, for example, we add a VCF. Now this VCF is a very standard GATK VCF, which is of like 8 MB. So once we start ingesting it creates a case where I can, like, as you can see, the pipeline status is pending. We can start processing it. So the process begins. um, so while the processing takes place, I will very quickly run through the different features that are available. For example, we do have an option to like all applications, we have a documentation where the documentation page is very clear, walks us through the logic as to how we are filtering and prioritizing variants. And for the purpose of this, we're using Civic, which is an open source literature database. And along with that, there is also the local knowledge base as well, which we are utilizing, which contains around 6,000 variants. And obviously we've got the contact support in case if any customization or anything is needed. So the architecture currently is also provided. Of course, these are technical things, but again, if anyone wants to like get through it, the setup, the deployment, all of it is like included. While we have these things, we also have the idea of where we can do bulk processing as well. So we have kept that provision open as well. So no need to like process each and every case one by one. So yeah, let's very quickly see what is the status of the case. As you can see, it is still processing, considering it's a big file, and the two steps, not just annotation. Okay, yeah. So since this is completed, we'll very quickly now we can review the variants. So, and as you can see, the number of variants that we had initially given was close to 29,000. Hence, obviously, it is taking a while to display the results as well. And we have also, since annotation, we figured out which of these are not quite relevant transcripts that are not having a lot of impact. So we get rid of them. We only retain the ones that are either having a high or a moderate impact. And along with that, obviously, we did filter out population variants as well. And as we can see, we've got all the different tiers. We can search for genes. We can also set the um, somatic allele frequency based on our choice. And obviously we've got different tumor indications. So for example now, so yeah, we can also look at them. So for now, for example, let's see the kind of variants that we have got. So we've got a lot of variants. And let's say we've got uh, yeah, we have selected those. Now what kind of information is available? So we've got the gene, we've got the cDNA, we've got the protein change, we've got the allele frequency. And from there we're looking at the different transcript types, the transcript ID, of course, and from there the consequence, also the population frequency along with the tier and level in accordance to the AMPAS-CoCap guidelines. And definitely we have got the different biomarkers types as well, which we can utilize in order to categorize variants. And of course the PMID, because evidence is what makes it important. So as you can see here in this case, we've got a hybrid. We have got data coming in from the local database. We have got data coming from the civic database as well. So that sort of like, yeah, some variants where both are available, so that is also provided. Now, with that being said, I'm just going to say click I'll proceed with these 11 variants. So obviously we have got all the variants. For example, and we can also drop them if required. For example, here, let's say for now, let's just retain like one BRCA2. We drop the rest of them. So for example, I'm just gonna drop them. So not integrating a lot of BRCA mutations, retaining only one of them. Along with that, we have got like different mutations in the dicer gene, IDH, as well. Lot of variants. And let's say given all of this, we now do the LLM annotation. As you can see, these are currently empty, which will get generated once I start generating the draft. Generate the LLM response. Now as you can see, this is currently starting to utilize the CPU as well as the memory. And as you can see, the usage starts speaking. And yeah, given this, the LLM response is starting to generate, which will of course take a while, considering this is not a PC which is powered by a GPU. Of course, on a GPU system, it's going to be magnitudes faster than it is on this currently. So yeah, as you can see, the information is generated. Now we'll also see, since there was a lot of variants that we queried, so that was around seven variants. Now hence for all the seven variants, it has taken what it's generating at the rate of 440 tokens per minute, which is quite decent compared to a PC which is performing on a completely on a generic CPU. And what we can do is, the crux of this is to download a report. Now, very quickly, let's see the report as well. Now, currently, as this is for short variants and doesn't accept any identifiable information, we have provided placeholders for the end user to like change things as well. And we also have like variants of, like, for example, there was no tier 1 variants detected, so this thing goes away. And accordingly, we've got tier 2, a lot of variants in tier 2, we've got those. And of course, since for all clinical potential clinical significance, that is all for all tier 2 variants, we have a gene summary which is generated by the LLM. We have got the variant summary, and of course we have got the evidence as well. And this is how it is set up. And yeah, with this of course we have got, based on all the information that is present, we eventually do get the references as well once we have to review, including the drug summary and all of it. So that is also provided by the engine, which can be used directly. And yeah, that being said, this is just the generic idea of how things look like. And yeah, I guess that's about it. That's about it. What we can do is, now once we're done with our work, we can always save a draft so that we don't have to generate the LLM stuff again and again. And we can always we can close the review, and we're back to the same page. And of course, we have got the option to delete things. And once we delete, obviously these get permanently deleted, including the VCF. So there is no footprint of these things whatsoever. And yeah, and if we need a log out, all you have to do is click terminate and you're done logging out. I guess that's about it, guys.